Okay, so I'm giving 2 lectures on basic mouse genetics. So why use the mouse as a model organism? Well, there are a number of practical, pragmatic reasons. It's easy to breed and house. It's easy to control the environment, so we can control the temperature that the mice are at. We can control what they eat. And we can control what enrichment they've got, how much exercise they do. There are physiological similarities with humans. A lot of the physiological pathways between mouse and human are highly conserved. And particularly in my own field of obesity and diabetes, that's particularly true. There are well-developed physiological techniques for the mouse. And there are genetically homogeneous strains available, so we can control the genetics in the mouse. There's a genomic sequence for 28 strains in the mouse, and I've put the web link there. I think you're getting these slides at some point, or they're going on the website, so you'll be able to follow these links. The mouse genome is readily manipulated. I think over the next few days you'll hear about some of the other techniques for manipulating the mouse genome, but we can make changes to the mouse genome and look at the effects of those changes on the physiology and the phenotypes of those animals. There are some things to be aware of in using the mouse as a model. The physiology of the mouse and the phenotypes that you measure is very strain dependent, so it matters what genetic background you're on or how mixed your genetic background is. Mice reach sexual maturity about 4 weeks of age. They have a rapid estrus cycle, so another consideration when you're doing experiments is if you're looking at female mice, you may have hormone effects that you can't control very well. Gestation's around 19 to 21 days, depending on the strain. Litter size is up to 10 pups, although I think we get somewhat less than that in our production colonies. And there's lots of information out there on the basic physiology of the mouse, all online and readily available. So if you need to look something up, or you want to know something, or you want to see what's known about the animals that you're working with, have a look online, and particularly the Jackson websites, Jackson MGI website. So I put a few links in for where you can find some basic data. So there are weight curves at the JAX lab. There's phenomic data, so that's phenotype data at the phenome at JAX and also at Europhenome. So if you want to look at traits and phenotypes, for example, there's some bone mineral density data there for a comparison between C3H and black 6 mice. And I'll explain a bit more about the strains shortly. So I said that the mouse is readily manipulated and that we've got genome sequence. So what do we know about the sequence of the mouse genome? This is another important source of information, the Ensemble website. And this is just some summary statistics on the mouse genome. So there's about 3.4 times 10 to the 9 base pairs in length. And this golden path is the assembled length of it's slightly shorter because there's lots of repetitive DNA and other things that are hard to order. There's somewhere around, you'll see this number sort of goes up and down, but there's somewhere around 22,000 coding genes, 11,000 or so non-coding genes, 8,000 pseudogenes. So these are cDNA sequences RNA sequences that have been retrotransposed into the genome, and so they don't have any introns but don't really function. And around 103,000 gene transcripts. So the mouse has 20 pairs of chromosomes. And one set of those is the XY pair, or XXXY. The longest chromosome is chromosome 1 at 197 megabases. These are recombination lengths, and I'll explain this a bit more shortly. So about 98 centimorgan recombination length. And chromosome 19 is 61 megabases long. And again, you can find this sort of information on the JAX website, which is where this picture comes from. The other thing to notice about mouse chromosomes is that they're acrocentric, so they have a centromere at one end. Whereas human chromosomes are dicentric, so they have a centromere between two arms. I'm going to give you a little bit of information on mouse-human comparisons now. So I've shown you that we've got lots of information on the mouse, phenotype data, genome sequence. But how good a comparison is there between mouse and human? And there's a little bit of terminology on this slide for describing mouse-human comparisons, taking the globin gene as a comparison, as an example, rather. So ancestrally, there's a single globin gene in lower organisms, but there was a gene duplication creating alpha and beta globin genes. So orthologs are genes in different species that come from a common ancestor. So the alpha globin gene, for example, in mouse, human for object, that's the ortholog. We refer to the ortholog in each of these species of the alpha globin gene, beta globin gene, for example. Paralogs are homologous genes within a single species that have diverged by gene duplication events. So there was a single globin gene, and now there's an alpha and a beta in the mouse, an alpha and beta in other species. And then homologs are all the globin genes, so anything that has homology with globin is a homolog. So that's just a bit of terminology. So mouse and humans separated some 90 million years ago. And there's been quite a bit of rearrangement of the chromosomes. Obviously, the chromosome number is different. And there's been quite a bit of rearrangement. So the colors here indicate mouse chromosomes. There should be 23 chromosomes here. I think there's something slightly wrong with the numbering. 23 pairs, so this is the human chromosomes. And you can see the colors indicate bits of mouse chromosome. So you can see there's been quite a bit of rearrangement, but within large blocks. So let's choose a color that I can actually see. Well, within this, I think this is a bit of chromosome 4, not sure. There's quite a lot of conservation of the order of genes, and then there's a break, and then there's a bit more of this chromosome ancestry somewhere else. So there are long stretches of DNA where the gene order's conserved between mouse and human, but then obviously they're not completely conserved as and organized as human chromosomes are. But there is good, I'm trying to make the point here that there's good defined synteny relationships between mouse and human. So this is some information from a recent PLOS biology paper. So we've already defined orthologs, and about 75% of mouse genes are in one-to-one orthologous relationships with human genes, and they show quite high amino acid and sequence homology, so around 85 to 88%. So there's good homology between mouse and human. The mouse has undergone more duplications than these are duplicated sequences in human and mouse, and there are more duplications in the mouse. And a large proportion of these are, in the case of the mouse, this has occurred because of the need to develop more sensitive senses of smell, for example. So the genes of olfaction, the genes involved in the sense of smell, are much more highly duplicated in mouse, than more dependent on smell than in humans. So this is a difference between mouse and human. And there are a number of other classes of genes where this is also the case. So genes involved in reproduction, for example, and a few in immunity, but mainly reproduction. So I thought I'd just say a few words, or I've got a few slides on the Mouse ENCODE project. So this is some stuff that's just been published recently. They're all open access publications, so you shouldn't have any trouble downloading the PDFs to read. There are actually 4 papers. There's these 3 papers. This is the overview paper, the Comparative Encyclopedia by Fang et al. And then there are some more detailed analyses on transacting circuitry and regulatory information. And then there's a 4th paper on DNA replication. Glauca has the issue there. So I thought and there are websites where you can look at this information as well. So there's the Mouse ENCODE, and then there's the ENCODE project. So this is an encyclopedia of DNA elements. So the ENCODE comes from these red letters. So the aim of this project is to map functional elements, well, initially in the human. So looking at, for example, DNase 1 hypersensitivity sites. So DNA is packaged into chromatin. It's wrapped around histones. It's quite tightly packed. But when it's transcribed, it has to be unpacked a bit. And when that happens, nucleases like DNase 1 can get in and cut the DNA. So you can identify transcriptionally active chromatin, DNA, by where DNase 1 can get in and cut, because that's what's necessary for transcriptional activity. The chromatin has to unwind, and that makes it vulnerable to DNase 1 digestion. So a lot of this data is maps of where DNase 1 sites are and what the sequence of those DNase 1 sites are, and those sites are correlated with transcriptionally active chromatin. But they've also looked at other levels of regulation in the genome. So you might have heard of DNA methylation. That's correlated with gene regulation as well. So DNA methylation on a genome-wide scale, they've mapped a lot of that. That data is on these websites if you want to look at it for your favorite gene. Chromatin modifications, so histone modifications, a lot of regulatory modifications through methylation of histones and acetylation of histones, and they've looked at those things as well on a global scale. There's rather a lot on here, but they're just the main points from the overview paper. And I've put this up because it's an interesting comparison between mouse and human, and there has been some talk about these papers in these sort of online discussion groups that this might imply that the mouse isn't a good model, but it is, and it's not. It's a misreading of what these papers actually say. So there's a lot of conservation between mouse and human, but there are differences in the expression profiles of many genes involved in various biological pathways. But there's been 90 million years of separate evolution. There's quite a bit of divergence of the regulatory landscape around genes. So the promoter elements on genes, the bits of DNA that control transcription, have been rearranged. Some of the factors of the transcription factors are the same in some species, in human or mouse. There are additional factors co-opted. So there is a certain amount of divergence at that level. However, the transcription factor networks are more highly conserved, and the chromatin state around these genes is more highly conserved between mouse and human. So it seems to me, at least, that this suggests that these papers support the mouse as a model. There are differences between mouse and human, and there are differences in the way genes are regulated. But at the sort of at the higher level of gene networks and gene controlling phenotypes, a lot of that is conserved. So I took out a few of the slides just to illustrate the sorts of things that they've been doing. So this is from the Stragarkis paper. So as I said, they were looking at mouse tissue. So they've done all this in human already with cell lines and tissues. And this is an ongoing project as well. And so they looked at around 25 mouse cell lines and different tissues. They looked, in this case, they're just looking at DNase 1 hypersensitivity sites, as they explained. The active chromatin is cuttable by DNase 1. And so they've sequenced these, they've identified these sites, they've sequenced them. They've got huge numbers of footprints across the genome that they can then compare with the similar data in humans. And if we just look at this chromosome 7 example around this gene NAPA, they've looked at the DNase 1 hypersensitivity sites, and it's quite noisy, but there's quite a bit of cutting anyway, but you can see conserved transcription factor binding sites. So these are the names of the transcription factors, so the gray lines in here. There are some that are not, but quite a few that are. So there is quite a bit of conservation there. And then in this final bit of the figure, they take these 8.6 million footprinted DNA elements for which they have the sequence, and they align them with human. So quite a few, a significant proportion have no matches in the human sequence, 35%, but 65, is it 65? I can't read it from here. 65% do. But when they look in their cell lines and tissues, only about 22% appear to be occupied in these footprints appear to be occupied in one of these cell types as well. So there's a large proportion that aren't footprinted in humans. So there's a mixed story here of conservation and differences, which you need to be aware of if you're using the mouse as a model, and in particular if you're looking at transcriptional networks, as we are. So that's just summarizes some of the data from that Stregarkis paper. So the 65% out of 65% of transcriptional factor footprints localized to the human genome sequence, only 22% are occupied in the human cell type assays occupied with the transcription factor. The actual DNA recognition sites are little change between mouse and human. So the transcription factors and the sequences that they bind to, well, they call it a lexicon, so it's a sort of dictionary of sites, for interpreting, for controlling gene expression, remains quite highly conserved at that particular sequence of the recognition sites. But there's divergence, as I've already alluded to, in the individual transcription factor sites being used. So here in the mouse, for example, a gene called gene A is regulated by three or more transcription factors. In the mouse, transcription factor 2 is unique to the mouse. In human, transcription factor 1 appears to be unique to the human, but they share some transcription factors in common. So there's a conserved connection between transcription factor 3 in mouse and human and gene A, although in this example, the sequence isn't conserved, so they're in different positions of the promoter. And transcription factor 4 is conserved but between mouse and human, but it also binds to the same sequence. So about half about 50% of the mouse regulatory interactions have this sort of conservation, conserved connections, and a proportion are conserved at the sequence level. And in this summary slide, then we can see individual DNA base pairs diverge the most. So transcription factors may be sticking to different parts of promoters. The individual footprints are conserved at around 22%. The connections between transcription factors is around 44%. So transcription factors controlling a gene are conserved around the 44% level. But the regulatory network architectures are even more highly conserved. So at the higher level, which is the point I was trying to make at the beginning, there is quite a lot of conservation. Oops, sorry, I skipped one. So I think I'll just round up by the point I wanted to make. The other point I wanted to make was that, for those of us who are looking at genes that have been mapped in human genome-wide association studies, one of the reassuring things that came out of these papers was when they looked at the candidate genes within these loci associated, for example, with diabetes, obesity, hypertension, whatever. They found that the distribution of the polymorphisms in the DNA that mark these genes are quite conserved between mouse and human. So there's, that supports the use of the mouse as a model for modeling those sorts of genes. So that was a bit of a divergence. Hopefully that wasn't too, but I suggest, look at the papers. They are quite complicated, but I just wanted to have a brief excursion into ENCODE because it's an important resource. It's all there, available online. You should be accessing that sort of information to look at your favorite genes. You need to think about the implications of those things, but it's a lot to take in one go. But the purpose of that little section was just to get you thinking about it and encouraging you to look at the available resource. It's all publicly available data, publicly funded, publicly available. So, make use of it. So I'll come back more to the basics now, having gone a bit non-basic. So inbred strains, what are they? They're maintained by brother-sister mating. They are genetically homogeneous, so every individual should be genetically identical. They're genetically standardized, so if you buy your mice from a reputable supplier and it says they're C57 black 6N, they should be the same as your mice you're using in your lab should be the same as the mice being used in somebody else's lab across the other side of the world. There are risks of, which I'll come to, of strains not being quite what they're advertised as, and of drift. So if you're maintaining your own colonies, you need a strategy to make sure that they stay homogeneous, and you need to refresh every now and again from a supplier. So different inbred strains will carry different polymorphisms. They'll have slightly different DNA sequences. They'll accumulate mutations. And they may exhibit different traits and phenotypes as a consequence. So don't assume that it doesn't matter what your strain is, because your strain may vary compared to other strains for the phenotypes that you're interested in. So blood glucose levels, for example. So as I've said, variously genetically identical, phenotypically very similar, homozygous at all gene loci. And these are some slides that I've pinched from a course that they run at Sanger that Sarah Wells, the director of the Mary Lyon Centre at Harwell, teaches on. And she updates them every year, and this hasn't changed so for the last year. So they're generated by brother-sister matings over long periods of time to homogenize their genomes. Some lines are lost due to inbreeding depression. So where do these inbred strains come from? They've been going for over 100 years now. Well, some of the lines have. So the origins of mouse genetics and inbred strains dates from around 1900. Originally, they were bred because people wanted to collect mouse fanciers, were interested in coat colors and having different coat colors and showing them, I guess, but also as pets. So Abby Lathrop began to collect these strains in around 1900 in the US and carried out fairly large-scale breeding experiments. And her mouse breeding operation was near the Bussey Institute, which was directed by William Castle, and she was their mouse supplier. So some of the common inbred strains, this is where mouse genetics began, some of the common inbred strains originate from these stocks. This book here, Lee Silver's book, is out of print, but you can buy it still on Amazon. And it's a very good it's a basic introduction to mouse genetics. So mouse genetics hasn't changed. the basics haven't, at least. So if you can get your hands on that book, it's quite useful. And I think they have it online. Well, they did it. I think they have it online at MGI, if you search around for it. So William Castle brought the fancy mouse into the laboratory around 1902 for investigating inheritance and variation, and his students, Clarence Little, developed some of the first inbred strains, and this line, DBA, which stands dilute brown non-agouti, was generated about 1909. In 1918, Little moved to Cold Spring Harbor and developed some of these other well-known strains, Baobab C, Black 6, Black 10. And then Little was involved in founding the Jackson Labs, so one of the big labs still involved in mouse genetics in 1929. There are different subspecies of mouse, depending on where they're from different parts of the world. So Mus musculus in Eastern Europe, Mus domesticus in Western Europe, Castanea Southeast Asia, Molossinus in Japan. And the inbred strains have been analyzed using high-density single nucleotide polymorphism genotyping. So looking at the DNA that they've inherited, looking at polymorphisms using these SMP markers to ask the question, how related are the inbred strains to the genotypes in these different wild mouse species? And the answer is that the classical lab strains are mainly domesticus, Western Europe, with a small amount of musculus, about 5%, and a little bit of castaneus. So these inbred strains have different names, and there is a genealogy that's published in Nature Genetics in 2000 that sort of tries to trace the relationship of all of these inbred strains and the sort of family tree for these animals. There are probably well over 450 inbred strains of mice described, providing a wide range of sequence variation and phenotypic diversity. This is from that paper, which comes with a poster. And here's one of our favorite strains, C57 Black 6. And you can see how it's related to other C57 Black 6 strains around the world. It's a bit small, I know, but it shows you that it's quite a complex family history. So it's important when you're using mice to know what your strains are and what their name is, and to use their name. And to when you write a paper on your data, make sure that you know what. Make sure that you report what strains you're using and report them accurately. So again, this is pinched from Sarah Wells' Managing Mouse Colonies talk. As I've said, there are all of these inbred strains, and they differ in various traits. So they have different coat colors, for example, agouti, and the agouti gene has the symbol A. Non-agouti, it's lowercase a, and agouti white belly, which has a white spot on its belly. And obviously the mouse, or all mammals, have 2 alleles for every gene, so you write a genotype like this. So the agouti mice are AA, and these black mice are A, small a over A. So and this is a single gene. So these are alleles of the agouti gene. So there's a gene called the agouti gene, makes a protein, controls coat color, and there are different versions of this, alleles of this gene. So agouti, non-agouti, agouti white belly. So I've explained the inbred stocks to you. There are other sorts of stocks. There are outbred stocks. So these are deliberately bred to try and maintain heterozygosity. So to try and make stocks that are more genetically polymorphic and perhaps represent what a human individual might be like. So sort of going against the whole inbred stock. The advantages of inbred strains is they're genetically homogeneous. The advantage of outbred stocks is they're not genetically homogeneous, so if you're testing a drug, then maybe you have a better chance of knowing if it's going to be efficacious or not when you put it in a really dirty genetic environment. There are transgenic, so these are genetically modified animals. So there may be gene knockouts or overexpression alleles where the genome's been manipulated. And there are mutations, point mutations, deletions, insertions, inversions. So there are stocks that carry mutations. So genotype, the mutations, polymorphisms that an animal has, interacts with environment. So as I said, we can control the environment to some extent, caging, enrichment, age, diet, temperature. And these interact to produce phenotypes, which are things that you can measure. So behavior, clinical chemistry values, for example. Anything that you can measure is a phenotype. But it's the interaction of genotype and environment. So it's important to be aware that you may have genetically homogeneous mice, but unless you're feeding them the same diet, or if you're trying to compare your study with somebody else's, unless you're feeding them the same diet and have them in the same environment, you may not get exactly the same phenotype. So phenotype's dependent on the interaction of genotype and environment. Oh, we've already done that one, sorry, it got duplicated. So inbreeding over long periods of time with lots of generations of inbreeding generates highly identical mice, and mice that don't vary genetically. So after 20 generations of inbreeding, 98.7% genetically identical, but over 150 you start to trend towards 100. So those will be isogenic, genetically identical, or almost. But there are spontaneous mutations, and these become fixed in the genome. So, co-color mutations, for example, are an obvious visible thing. So, how do you protect yourself against genetic drift? So, if you've bought your mice in, you've kept them for 10 years, they're going to change because they're going to accumulate mutations, assuming that you haven't contaminated them with another strain. So there are various strategies for doing this. Breeding strategies. You can cryopreserve, so you can freeze sperm from mice relatively straightforwardly now, and there are courses in various places, including at Harwell, where you can learn to do this. So you can secure your stock by cryopreserving it and then going back to that, so refreshing your stock every now and again, or you can buy in mice to refix and let somebody else worry about maintaining the stocks and the integrity of those stocks. I think I'll skip this one. So these are this is the more complex schemes that the Jackson Laboratory, who's a reference supplier of inbred strains and genetically modified lines. So you need to be able to, so in managing your colonies, you need to be able to identify individuals, so there are ear-coding schemes that you can use, and there are other more sophisticated ways of tracking mice like chipping. You need fairly strict husbandry regimes, so it helps to have different racks for different coat colors or different mutations. Don't handle them at the same time in the same place, or the same time or in the same place. Watch for deviations from normal, because you will get drift. So there are various sort of procedural things you can do to try and maintain the integrity of your stocks. So there is genetic drift. You do get mutations. They can be difficult to detect. They can be invisible phenotypically. So one rule of thumb is if a colony has been separated from its parent colony by more than 20 generations, it should be considered a new substrain. So you'll see this is the C3H strain. So this is the name of the parental strain. And HEH refers to the laboratory that this substrain is now in. So you'll find there are lots of C3H substrains, and they usually have they will have a laboratory code on them as well. So, here we've got 6J, which is Jackson, 6N, which is NTAC, which is Taconic, I think, that one. So, and they do differ, which I think will be on a later slide. So, as I said, the cause of genetic drift is that there are mutations, and you're inbreeding your stock all the time. Some of these will be lost. So with further inbreeding, this mutation will just disappear, be bred out, won't be retained. But sometimes these mutations will become fixed, so they'll end up being homozygous, and then they're there forever in the stock. So that's how they drift. Get a mutation, it may get fixed over time. So you'll end up with a different strain to the one you started with. I mean, the other risk is genetic contamination. So if the wrong mouse gets in the cage, if you're dealing with thousands of mice, these things can happen. So you need strategies in place to look for genetic contamination if you're operating a colony, if you're a colony manager. And as I said, we know the sequences of these strains, and we can genotype them with single nucleotide polymorphisms. Maybe one day we'll sequence them when it becomes cheap enough. So you can look at the integrity of your strain by just running a genotyping panel, looking at the bases, or looking at the single nucleotide polymorphisms, and look for differences. So that they should all be in a particular stock, they should all be the same. And if they're not, then you have a problem. So you can test so you should be able to identify whether or not you've accidentally crossed in another mouse strain. And in which case you go back to an earlier frozen individual, or you refresh your stock from outside. So you can detect gross contamination from another strain. Obviously, there's a limit on the sensitivity of this. And if it's a small bit of DNA, you might not see it. But the likelihood is, if it's a completely different strain, you will see it. But if it's a transgenic that's got a little bit of something else left in it, you might not. So, as I said, spontaneous mutations occur at about 1 in 3 times, 5 times 10 to the minus 6 per gene per generation. So after 16 generations, there's a 90% chance that 2 substrains will differ at 1 or more gene loci. So it's going on all the time. It's not something you have to live with, and something you have to guard against. So this is coat colors. This is an obvious phenodeviant in that they have these black and white splotches that are segregating in the colony. We had one in a colony it was a black 6 colony actually, and it's a quite you don't know about this mutation until you look for it. We were looking for thermoregulation mutants, so we were doing a cold challenge on these mice, and so we were chilling the mice and following their body temperature over several hours, and we found that the background strain that we were using, or at least some of the mice in the background strain, had this more rapid decline in body temperature than the other mice in the colony. so we were able to map this, and we identified a mutation in the uncoupling protein 1, which is involved in brown adipose tissue and generating heat by non-shivering thermogenesis. So this was a UCP1 mutation that arose in our black 6J colony. We didn't know about it until we looked for a thermoregulation phenotype, but it was there. It wasn't fixed because not all the animals had it, but we had to then go and clean it out of the colony. And that was the only mutation we found in that screen, which is a bit sad, because we wanted novel things, not UCP1. It was a good positive control. But, and this is just another example, so I think we won't worry about these. So they can be useful. These mutations can be useful, so you might find that they're interesting, like the UCP1 would have been interesting if it was a novel gene, but it was already well characterized. But various inbred strains do carry mutations. So, for example, the NNT gene in C57 black 6J, it carries a deletion that reduces insulin secretion. But that deletion is not in 6N. So if you've got C57 black 6J mice, they'll have a deletion in this gene, and they have reduced insulin secretion. If you have C57 black 6N mice, which were separated from 6J, they don't have reduced insulin secretion, so they don't have a glucose tolerance phenotype. So as I've said, obtain your mice from a reputable source, Jackson Laboratory, Charles River, for example. There are other suppliers. Maintain good records so that you know what a mouse is and what its relationship to the other mice in the colony is. It's important to keep those sorts of records. If you can, cryopreserve embryos or sperm every 10 generations and replenish your colonies from the original source, because after you've kept them for 20 or so generations, you've got your own strain then. As I've said, phenotypes differ on different inbred strains, so this leptin receptor mutation, which causes mice to be very obese, on a 6J background is milder than on a KS background, where you get obesity and overt diabetes that will kill the mice, whereas on a 6J background, it's milder. I've already told you about the 6J, 6N difference in glucose tolerance, and this is actually data that's from UConn. And there's a genome biology paper comparing 6N and 6J, and this is one of the traits that they found was different. How are we doing for time? Okay.
You finished the hour. You want one question in between, or?
We could do, yeah. I've got a few more yet, but I'm just checking. I'm not going too slowly. So outbred strains, closed population, genetically variable animals. These are useful in some contexts, as I said, if you want a particularly diverse genetic background, although you need to question how diverse those backgrounds really are. But again, you can buy these things. And that's probably the safest way to do it. So, toxicology, pharmacology, these things are particularly useful and as foster mothers because they're very healthy mice. So if you bring a mutation in, and it's on it's to be on a particular genetic background, or it might be on a mixed genetic background, you can cross it onto a background of choice, so you can standardize it, so you know what you've got, and it's on the background you want. But this takes time and it costs money because somebody has to look after the mice, and you have to do lots and lots of back crossing. So you have a number of other options. So you can buy it on the right background or on a clean background. And your other option would be to maintain it as a heterozygous background. So if it's on a particular background and you want to change it, you could make an F1. So you have to, in order to maintain genetic integrity, you've got to be aware of genetic drift, and you've got to have procedures in place to minimize genetic variance and have good breeding practices. So that's the first section. So we can either take some questions or...
Yes. Thank you.
Thank you.